Y'all, Jack Schultz. I'm an undergraduate in Dr. Busby's lab, where my work focuses on plant-fungal interactions below ground, specifically that between the plant Pseudotsuga menziesii, Douglas fir, and the ectomycorrhizal fungi species, specifically the presence of this mutualism along an elevation gradient. This work is part of a bigger project being done by graduate student Abigail Neat within Dr. Busby's lab. Um, is there a clicker?
Click on the PowerPoint.
Okay, thank you, Andy. This relationship between plants and fungi is often mutualistic, playing critical roles in ecosystem functioning through nutrient cycling and resource allocation. It is believed that this relationship is nearly 400 million years old, with its development being cited as a critical component in the migration of plants on the terrestrial landscapes. The ectomycorrhizal form of the mycorrhizal relationship occurs specifically between a plant partner and a fungal partner in the clods of Ascomycota, Basidiomycota, and Glomeromycota within the kingdom fungi. This relationship, within it, the fungi acts as an extension of the root system, accessing organic nutrients and water that would otherwise be unavailable to the plant. This relationship occurs on the outer face of the root, where fungal tissue colonizes the exterior of the root tip. This is displayed in this kind of blue purplish tissue. This exterior layer is known as the fungal mantle. There's also colonization occurring within the cortex of the root. Here in between the epidermal cells, fungal tissue colonizes this empty space, and this is known as a Harding net. Here, where plant and fungal tissue connect directly, there are specialized aquaporons and transport proteins that allow for the fungi to directly allocate nutrients and water to the plant. This is what the colonization looks like under a microscope. Here we have a sectioned root tip that is colonized. This is a photo I took under a electronic microscope. You can see the fungal mantle tissue on the exterior. It's going to be this light, fluffy tissue. It's definitely going to be harder to see, but there's colonization occurring within the cortex of the root too. This is how the mantle appears under a dissecting microscope. This is not, how a normal root tip looks. There's going to be a distinctive color and kind of morphology. This relationship is not, charity. The plant reciprocates with photosynthesized derived carbohydrates or these long sugar chains. There's usually a cost benefit, carbon cost benefit associated with this relationship that's not fixed and is reliant on numerous factors. One of the largest being abiotic stress in an environment. This is going to be climactic pressures present. And according to the stress gradient hypothesis proposed by Dr. Mark Burtness, environments with higher levels of abiotic stress are going to see an increased mutualism because of the benefits provided by the mycorrhizal relationship. This is going to be provided resilience towards these stressors as well as an increase in resource allocation that's really going to outweigh the carbon cost of this relationship and kind of justify its presence within these ecosystems. However, in habitats with low abiotic stress, where nutrients are readily available and there's not much pressure, this relationship is really not going to be justified through the cost. And so here we'll see a decrease in the mutualism present. We're curious to how this will look within the temperate rainforests of the Pacific Northwest, and specifically within the Cascade region of Oregon. This is a region that has numerous habitats and climates, each encompassing a various level of abiotic stress. This forest also is dominated by a common ectomycorrhizal partner, Pseudotsuga menziesii or Douglas fir. So because of this, we think that this is kind of the ideal landscape to explore how various levels of abiotic stress present in a habitat level influence the presence of mutualists in the plant root system. So we are curious. Or one proponent of stress within this region is elevation. Low elevation forests here are going to be very dense, thick forests with a lot of understory vegetation. They're going to have a pretty heavy canopy. And these forests can receive up to 2,000 centimeters of precipitation annually. These forests also have a pretty short freeze cycle. However, forests at high elevation within this region are going to be a lot more bare. They're not going to have as much, vegetation. They're going to have a pretty sparse canopy. These forests are going to receive less annual precipitation than their low elevation counterparts. and can have freeze cycles that last 3 to 4 times longer. Within the work of Dr. Busby, Dr. Jones, and Dr. Lamanna, exploring conspecific negative density dependence along a similar elevation gradient within the Cascades, they characterize these high elevation forests as encompassing a greater level of abiotic stress. And so through collecting seeds and soils from along habitats in this elevation gradient, we aim to collect organisms that reflect the uniqueness of these habitats. And then by analyzing mutualism presence within the seeds and microbes sourced along this elevation gradient, we aim to kind of look at how abiotic stress present within a habitat influences the mutualism presence. So here we're kind of curious to whether this stress gradient hypothesis will translate into an increase of ectomycorrhizal colonization abundance as you move up along the elevation gradient. This would result in the greatest amount of colonization being observed in seeds and microbes sourced from high elevation ecosystems. So here, in a climate-controlled greenhouse experiment, we see how the abundance of this ectomycorrhizal partner is influenced by local seed source, microbial communities, and drought from along an elevation gradient, which we hope serves as a proxy for an abiotic stress gradient. So to really explore this, we created 4 hypotheses. The first is going to be the seed source hypothesis. This is where we explore the impact seed source has on ectomycorrhizal abundance to explore if this plays an impact on the presence within the root systems. Here we hypothesize that seedlings from high elevation seed sources are going to have the greatest abundance of ectomycorrhizal colonization because they're originating in these habitats that we hypothesize as being more abiotically stressful. The next is this microbial source hypothesis, where we explore the impact that microbe source has on ectomycorrhizal abundance. Here we predict that seedlings grown in soils with high elevation microbes are going to have a greater abundance of this mutualism in the root systems. Again, because these organisms are originating from habitats that we believe are more abiotically stressful. This third hypothesis is kind of going to look at both of them within the interaction. This is going to explore local preference by analyzing microbial source impact on a seed source level. So seeing how each seed source responds to being grown in different soil treatments. Here we're really curious if there's going to be local preference, which would manifest in an increase of ectomycorrhizal abundance in seed sources when they were grown in microbes from their own elevation. Here we hypothesize that seedlings from high elevation seed sources grown in soils sourced from high elevations are going to have the greatest abundance of ectomycorrhizal colonization. And then lastly, we have this drought interaction hypothesis, where we go back through and explore all 3 of the first hypotheses using seedlings that were exposed to a month-long period of drought. And this is really to explore how applied abiotic stress is going to impact ectomycorrhizal abundance on a seed source level, a microbial source level, and then in their interaction. Yeah, and so to do this, we created a project with 3 seed source treatments and then 3 microbe or soil treatments. The 3 seed source treatments are going to be high elevation seed sources, low elevation seed sources, and mid elevation seed sources, with those microbe treatments being soils with high elevation microbes, soils with low elevation microbes, and then a third soil that it's just sterile potting soil that's supposed to kind of act as a control in this experiment. This results in 9 independent treatments, which is then doubled into 18 when that drought is applied to half the seedlings. Seedlings were collected from 6 sites along a 600-meter elevation gradient within the Oregon Cascades. Sites were paired so that 2 represent low elevation, 2 represent mid, and 2 represent high, with sites A and B being low elevation forests at just above 400 meters. Sites C and D representing mid elevation forests at just above 600 and just above 800 meters. And then sites E and F representing high elevation forests at just above 1,000 meters. Here at each site, premature cones were collected from mature Douglas Fir trees. Soil was also collected at the highest and lowest sites, sites A and site F. Here, soil was collected from a depth of 10 centimeters underneath 10 focal trees at each site. Soil was then brought back to the OSU greenhouses where it was mixed with sterile potting soil at a ratio of 1 to 16 to create the two microbe treatments, along with that third one, which is just sterile potting soil. Seeds were also, brought back to the OSU greenhouses where they were germinated and stratified, and then grown in either high elevation soils, low elevation soils, with a third smaller population being grown in that sterile control. These seedlings were grown in the OSU greenhouses where their trays were rotated every few weeks to kind of minimize the impact that microclimates have on these seedlings. After about a year of growth, half of the seedlings, about a population of 550, were exposed to a month-long period of drought in which they were only watered 50 to 70% full saturation, while the other half was watered to complete saturation. After this month-long period, watering intervals returned to normal, and a few months later, in September of 2023, seedlings were harvested from their pots, given a root wash, and then kept in -20 degrees storage. This is where I come into this project. I take these seedlings out of the freezer blindly to not look at the treatments. I don't want to create any sampling bias there. I then put these seedlings in a root wash to rehydrate any fungal tissue present and to remove any extra sediment. I then place the seedlings in additional root wash bath placed underneath a dissecting microscope. I then visually segment each root system into thirds. This is to ensure that the whole root system is present within the analysis, avoiding any sampling bias from a single part of the root system. Within each third, I randomly select 20 root tips and then score those based off of visual colonization. This totals in 60 root tips for each individual seedling. And then with roughly about 1,100 seedlings involved in this project, this then totals to a little bit above 60,000 root tips scored over a 3 to 4 month period. To score a root tip off of colonization, you first need to know what an uncolonized root tip looks like. These are going to be pretty inconspicuous little root tips. They don't, deviate and look. They're going to be, all over the root system, and they're pretty basic. They have an abundance of root hairs, a color that matches and texture that matches the rest of the root system. So a colonized root tip is going to deviate in three major ways. The first is going to be morphology. So here are the two main types of root tip morphology of a colonized root. First, we have a tight, highly clustered root tip morphology. We're going to call this like a tuning fork. And the next is a highly elongated and highly branching morphology. We refer to these as Christmas trees. Next is going to be the presence of fungal mantle. So this is going to appear as kind of a cottony, soft tissue surrounding a root tip. It's going to make it appear more bulbous, and it's also going to have usually a distinct color with it. These ones up here are a little harder to see. This is a colonized root tip versus an uncolonized, and that's going to have like kind of a darker iridescent gray color, whereas this colonized root tip right here is going to be a little more noticeable with a bright pinkish yellow mantle along it. So the third final kind of indicator that there is a presence of ectomycorrhizal colonization is going to be the absence of any root hairs. These root tips can have, maybe 1 to 2 root hairs on them, but they're really going to lack that abundance that you observe on an uncolonized root tip. So if you come across a Douglas Fir root that, has a funky morphology, has kind of a cottony tissue surrounding it, and lacks any root hair, then you know that that is going to be a colonized root tip. So using those cues, I went through and scored the root tips, which gave me a individual colonization percentage for each seedling. This data was then placed into R with the first 3 hypotheses, each consisting of a single model. And that last hypothesis, the drought interaction hypothesis, where we re-explore the first 3 hypotheses using droughted seedlings. This is going to consist of 3 models. So that totals to 6 total models. And then within the data, we used generalized linear models to look at the main effects and significance within the interaction. When significance was found, we used Tukey HSD pairwise comparisons to explore significance between treatments. We then used ggplot to graph the data. So now we're going to go and look at the results. So first we have this seed source hypothesis. And you can tell that there is, a significant difference between low and high elevation seed sources, as well as between mid and high elevation seed sources. This is paired with a very significant p-value. And our R squared value is, pretty low, which is going to be a consistent pattern within our data. We believe that this is going to be the case simply because of the size of our, the individual tree or individuals within this project. We're going to have a lot of outliers, which are visual in the data. So this is going to bring that R squared value pretty low. So really what we're going to rely on to distinguish, significance within the data is going to be a very significant p-value, which we do have in this case. We hypothesized here that we would, see an increase of ectomycorrhizal abundance as you moved up along that elevation gradient. However, this really was not the case. What we observed directly contradicted that. It showed that the greatest abundance of ectomycorrhizal fungi was really present in these low and mid elevation seed sources, with the lowest being found in that high elevation seed source. This directly contradicts our hypothesis. And one reason we thought this could be the case is because of historic mutualism limiters present at these high elevations. Dr. Lee at the University of Colorado found that in these freezing conditions, photosynthesis is significantly down regulated in Pseudosugum menziesii. These high elevation forests, can be under these freezing conditions from late fall to early spring. So this is going to be a significant kind of time frame where these trees can't, produce carbohydrates to their best ability. And this can really, limit the amount of carbohydrates they can spare for this mutualism. So, this could, conceivably lead to just historically a lower or higher limitation in the amount of mutualists that they can harbor within their root systems. However, low elevation forests, they really don't have this stress present. They might have these conditions for 1 or 2 weeks out of the year. So therefore, they're not really going to have this significant downregulation of carbohydrate production, conceivably leading to a higher ability to harbor mutualisms just because they have this increased carbohydrate supply. To actually tell if this is the case, we would need future research. And I don't know the best way of going about this, so if you guys have any ideas, I would love to hear them afterwards. Another reason we could be observing these results is because we came into this project assuming that high elevation ecosystems are going to be more abiotically stressful because of these long freeze cycles and lower precipitation rates. However, these aren't the only abiotic stressors that really influence ectomycorrhizal necessity and abundance. There's going to be a ton of factors that play into this. A lot of these are kind of explored in surveys of this region. One specific one is the Johnson et al. Survey of H.J. Andrews, an experimental forest within this region. Here he found that low elevation forests actually have lower nutrient availability, lower organic material, and then warmer seasonal climates. These forests get really hot during the summer. And so these are going to be unique abiotic factors that could really, create a demand for mutualists at these low elevation forests. Yeah, so going back through, unfortunately, we didn't, have any support for our hypothesis, but we did, in fact, see that seed source plays a significant role in colonization abundance. And this is really going to be a key takeaway from this project. Next, we're going to look at microbial source hypothesis. And here, unfortunately, we see that there is no significant difference between the data. That's a pretty high p-value, and then the R-square value is, insanely low. That really signifies that there is no difference between the data. And that's, weird because we're supposed to have this sterile control that's supposed to act as a, not have any microbes in it. But when that, that percent colonization of the sterile control is as high as these treatments with field organisms and field microbes in them, that definitely signifies that there was some sort of contamination. And when going through scoring these seedlings, it was definitely noticeable that there was a single type of morphology of colonization present within these root systems. And using previous knowledge from past projects within these greenhouses, we came to the conclusion that this is going to be a Telephora species, which is known to be pretty, pretty present within the OSU greenhouses. So future research would definitely want to explore, this on a different level to actually have, a sterile control. So here we hypothesized again that there would be an increase in abundance of these mutualists as you went up along this elevation gradient. But this really was not the case in our data. We saw that after a year, there really was no difference in abundance of mutualisms based on which soil seedlings were grown in. One reason this could be is simply because this project went on for only a year and a half. This in the large scale, this isn't a super long time. Research out of UBC by Dr. Twegg has found that as tree age increase and forest age increase, there's an increase in structure of the microbial community. So maybe if we allowed this project to go on for another 5, 10 years, we could really see a divergence in those colonization patterns on a microbial source level. However, for this project, we really didn't find that there was any support for our hypothesis, and we saw that after a year there wasn't a difference in the microbial sources. Now looking at the interaction hypothesis, we see that there is a very significant p-value and honestly a relatively high R-squared value for this data set. However, using the pairwise comparison, we see that on a seed source level, there is no statistical difference in microbial treatments. This means that seed sources usually had similar abundance levels, regardless of what soil type that they were grown in. This reveals that there is really no local preference visible, as you would kind of see here where we hypothesize kind of this like flip off of abundance, where lower elevation seed sources would have a greater percent colonization, and low microbes, which would also be true in high seed sources exposed to high elevation microbes. But this really is not the case within our data. We observe that any difference in colonization patterns are really on at that seed source level, kind of reiterating the importance that seed source has on colonization abundance. Yeah, so here, unfortunately, our local preference hypothesis was not supported. However, we did see that, seed source has a significant role in colonization presence. So again, this is going to be a major finding, a pretty big finding within this work. We'll now kind of move on to this drought interaction hypothesis, where we look at these first three over again, using a different set of seedlings that were exposed to that month-long drought period. So looking at it on a seed source level, results kind of look, pretty similar to our watered seedlings. We see that there is a very significant p-value and a relatively high R-squared value for this data set. We see again that there is a significant difference between mid and high elevation seed sources, as well as low and high elevation seed sources that kind of follow that same trend that we saw in watered seedlings. However, we hypothesized here that kind of this difference between low elevation seed sources and high elevation seed sources would kind of strengthen when drought was applied. However, this was not the case. We did see that there was an overall increase of ectomycorrhizal abundance across all treatments when drought was applied. However, that's not necessarily what we were looking for. We saw no difference in colonization patterns that would have indicated kind of a difference from the watered seedlings. And again, even though we saw that our hypothesis wasn't supported, we did see that seed source plays a role in colonization abundance. Looking at a microbial source level in dry seedlings, initially were met with a significant value between high and low elevation seed sources, showing that there is greater colonization in these high elevation microbes. However, our p-value, while significant, it's still fairly high, and then that R-squared value is going to be really low. And there's also again insignificance between that supposed sterile control. So because of this, we're really going to be cautious in interpreting these results as being a product of difference in microbial treatments. So again, here, we hypothesize that the difference between low and high treatments would strengthen when drought is applied, favoring these high elevation soils. However, this is not the case. Again, we did see that there was an overall increase in colonization when drought was applied. However, we can't determine that there was any difference in colonization abundance as a result of the different microbial treatments. For the drought interaction, again, we're met with a very significant p-value and the highest R-square value that we're going to get out of this data set. But But again, the pairwise comparison reveals that there is no difference between microbe treatments on a seed source level. This again, shows that there is really no local preference observable within this data. And, any difference that we do see in colonization, again, is going to be on that seed source level. So unfortunately, we didn't have any support for our drought interaction hypothesis here. But we did again see that, seed source origin plays a significant role in colonization abundance. So that's definitely going to be a key takeaway from this project. Seed source plays a big role. After a year, microbial sources did not play any kind of significant role in colonization patterns. And any difference that we saw in the data was on that seed source level. Future research would definitely want to explore this microbial source a little more. And a way to do that would definitely to create a genuine control, sterile source. A way to do this would use very sterile kind of growth chambers to create, a genuine control, which would allow us to kind of analyze these field microbes a little better. However, this would kind of limit the size of the project as these provide a lot less space than a greenhouse, and they're also less cost effective. However, another approach would be a field-based approach, such as a reciprocal transplant project. This would allow you to plant these seed sources directly into the field along this elevation gradient, and would give you better insight into how, genuine abiotic stresses in microbial communities present at these habitats would impact seed source differences. And so this would also kind of reveal whether or not our results are just kind of a novel product of the greenhouse environment, or if there is truly a difference in colonization on a local seed source level. When considering implications of this work, it definitely touches on drought, how local seed sources and microbe sources respond to drought, which is, an abiotic stress that's going to become more prevalent in these forests as climate change worsens. So knowing how, local microbes and trees respond to drought can kind of piece together how a forest as a whole will respond to drought, which is definitely something that's important to know in the ongoing future. Another implication of this work kind of touches on reforestation efforts. Since we did find that there was a difference on a seed source level, maybe, having numerous seed sources for a reforestation effort can create kind of a diverse and selective habitat that selects towards genotypes or seed sources that are better adapted to those microclimates. This kind of remove an element of homogeny from these environments and hopefully create a healthier forest. So yeah, I definitely want to thank the Busby Lab for all the help and support over the last few years, the Jones Lab, the Botany and Plant Pathology Department as a whole. I definitely want to thank, the Bioresource Research Department. Wanda has been such a huge help, Gail, as well as Kate Field. I want to thank definitely my parents and my friends for all their support. And lastly, the Sonoma County Mycological Association for funding for parts of this project. Yeah, thank you all. This has been exciting. I love it. Now field any questions that anyone has. If there are any. Yes, Haley. Thank you.
You were talking about this historic mutualism limitation in the forest, and I'm curious what your thoughts are on maybe weather patterns in the years preceding the seed collection for this project, and whether there were any major events that you think are relevant, and then also what you see in the future. I know you talked about drought, but just snowpack and temperature in those high elevation regions.
Yeah. I don't know any specific weather patterns. I do know that, kind of in the summers we've been seeing hotter climates, so this can definitely impact those lower elevation seed sources and microbe sources. However, is it becoming more, I know you live out there, are winters becoming more severe, or are they lessening? Okay, rainier. Yeah, so maybe these, historic limitations will, be decreased. I'm not quite sure of how. I also don't know how short term these changes in limitations would be. I don't know if it would be over a few years you would see a difference, or it would have to be over a few decades. However, yeah, that would be a good avenue to explore next. Joey.
Jack, on your microbial source hypothesis, we have the phyllophora that complicated things. So in your control, you had all phyllophora, right?
Yes.
Yeah. In your two treatments, did you see a diversity of morphotypes of root tips?
We did. We saw a lot of that pink yellow colonization, which we associated with rhizopogon. I believe we saw a little Cenococcum. We definitely saw that even in like the darker, kind of less distinguishable kind of morphotypes, there was a bigger diversity in these field treatments than the sterile treatment, which we really did only see in the sterile treatments, it was just kind of a gray, thick, kind of iridescent tissue, which we associated with the telophora.
And on the drought, I thought the drought data was really interesting. That's cool. Were there particular morphotypes and when you saw that increase in microization due to drought, were there particular morphotypes that you could attribute that to?
Definitely, I believe this kind of touches on previous work that Abby's definitely done with rhizopogon. It seemed like rhizopogon abundance increased when drought was applied. And we know that, rhizopogon is a species that helps a lot in drought resilience in these trees. So yeah, we saw definitely an increase in that pink tissue in these root systems when drought was applied.
Cool.
Any other questions? Yes, Wanda.
Along those same lines, is like, if you were to have looked at the number of different types, the diversity versus, or going the other way, which is looking at scoring it plus or minus, right? Did they have it? Did they not? And as opposed to percent, I wonder if you would have had a more cleaner breakout.
Yeah, going through and visually kind of doing that species abundance is going to be hard. That would definitely want to be something like you do metabarcoding for. I don't know how reliable visual colonization would be for that to know kind of the abundance of species. But one approach that we were going to take that Andy really wanted me to take is kind of plotting it on this logarithmic scale to kind of show the difference between the sterile soil and the field soils. However, because those sterile soils are, not really sterile, it wouldn't really give us a good idea of the influence that these field microbes have. So that would definitely be an approach that we would take in future projects.
I was just thinking that the plus or minus, is it there or not? If you're a microbe, how long is it going to take you to get up to speed if the conditions are right? You're going to take advantage of that, right? Did you notice any difference in the size of the seedlings in terms of whether they were colonized or not, or?
Definitely. That's a big element of Abby's work, is looking at the more physiological side of that. I know that there was definitely kind of a trade-off in colonization versus size. I believe that we saw that when seedlings had greater levels of colonization, I believe they were smaller. And so there is definitely an observed trade-off on size. We don't know how that affects necessarily fitness. We would assume that these colonized seeds would be more fit because of the provided resilience and nutrients that they get. But they also have this trade-off of supplying carbon to the fungi, so that could limit their growth really. But yes, we did see a difference, and that can also be a big part of future research is adding that physiological side.
Any other questions?
Let's say yes.
Thank you. Do you think that there's any differences for your seed source hypothesis, like in the seed, the microbes that are in the seed already? Or has any work been done to look at?
I'm not sure. Yeah, that would be great work. I do know that the cones were, I think, were they sterilized when we got them from the field? Okay.
We sterilize the seeds.
Okay. So seeds were sterilized, so I'm assuming that would kind of minimize that effect. But again, that would be a really cool project to explore unsterilized versus sterilized seed sources. Yeah, I suppose you.
Talk a little bit about whether you think that the seedlings were nutrient limited in this experiment and the degree to which the mycorrhizal fungi, where they sort of were in this sort of mutualist to parasite continuum.
Yeah, so the soil was fertilized. I believe this is potting soil. So I don't believe that there was a huge limitation in nutrients there. I mean, am curious of how that plays out because that's not the case, in the field. And so usually these seedlings aren't all exposed to the same level of nutrients. So that's definitely something I considered this, spectrum from mutualism to antagonist. Um, and kind of going off of that, I definitely assume that maybe these lower elevation ecosystems that could have less abiotic stress might have a greater presence of these maybe non necessarily commensal ectomycorrhizal partners. However, that would take metabarcoding and PCR to kind of attribute a level of parasitism or mutualism to these fungi. So that definitely would be a good avenue, but it would take a little more, I think, genetic analysis to determine who's actually there. Yes. All right.
There are no more questions, then let's give Jack a round of applause. We're going to transition to the next portion of Jack's thesis presentation. So Dr. Jones, Dr. Busby, self and Jack will talk about the work that you've done. Take time to break if you need to. I'll shut down Zoom and get
Did
us
you
set
check
up.
if there's any questions online?
Oh, no questions online, but congratulations, Jack, for your talk. Thanks for joining us online.
Thank you, everyone.